My name is Niccolo Zanichelli. I'm an undergraduate student in computer science at the University of Parma, Italy, and one of the organizers behind OpenBioML. Today's presentation is about artificial intelligence in computational biology and the increasing relevance of scale in this context. Let's begin. We will begin by discussing some of the most important recent developments in machine learning when applied to protein structure prediction and protein design. First, however, let's briefly discuss what proteins are and why these applications are so important. Proteins are large and complex molecules that form the basic building blocks of life. They are made up of amino acids and are responsible for nearly every task of cellular life. Each protein is characterized by a specific sequence of amino acids, which gives rise to a corresponding three-dimensional structure that the protein naturally folds into. A protein can thus be described in multiple ways. The primary structure of a protein is its sequence of amino acids. The secondary structure of a protein is its local conformation and local structure, that is, the so-called alpha helices and plated sheets that characterize it. The tertiary structure of a protein is the full three-dimensional structure associated with it. A protein's tertiary structure has a tangible impact on its function and often determines it completely. It is for this reason that protein structure prediction, that is, the problem of predicting the three-dimensional shape of a protein given its sequence of amino acids, is an extremely important problem in computational biology. A true solution to the problem would effectively provide us with the ability to design proteins with any desired structure and function. This could have transformative impact on a number of downstream applications, including drug discovery, synthetic biology, climate technology, and much more. Let's take a look at some of the most important recent developments in machine learning for protein structure prediction then, over the last few years, beginning with DeepMind Alpha2. I won't go into too much depth about the model, as AlphaFold 1 is from 2018 and its influence on the field is somewhat limited in comparison to the second and more recent version. But there are a few notes worth making. AlphaFold 1's input is composed of two important features, the amino acid sequence of the protein and a set of multiple sequence alignments, which are essentially a representation of the evolutionary history of the protein itself. The core of the model is a series of convolutional residual blocks. These blocks, however, have a limited field of view and are not able to integrate all information available to the network. This is a key limitation of AlphaFold 1, and as we will see later, the resolution of this issue led to a large performance improvement in AlphaFold 2. AlphaFold 1's output is a distance matrix, which quantifies what the model believes to be the distance between every pair of amino acids. This distance matrix is used as input for a process based on gradient descent, which minimizes a set of protein family-specific energy potentials and attempts to find the resulting tertiary structure. The training of the model is not end-to-end, as the model is only differentiable up to the pairwise distance matrix. This is another key limitation of AlphaFold 1. In the right of the image, it's possible to see an illustration of how the protein is folded during the gradient descent, starting with the structure predicted by AlphaFold 1's pairwise distance matrix, and ending with the structure obtained. AlphaFold 1 participated in Casp 2018, which is the most important competition and benchmark for protein structure prediction research. And in the image, you can see its performance in comparison to all other participants. Without going too much in depth about the specific metrics used, the key point to make is that, as the image shows, AlphaFold 1 was a significant improvement over the state-of-the-art of the field. Soon after the DeepMind announced the results of AlphaFold 1, Facebook AI, now Meta AI, developed ESM, Evolutionary Scale Modeling, a family of protein language models based on a concept similar to BERT, the famous language model architecture from natural language processing. The main idea behind ESM is to leverage an analogy between natural language and protein sequences. In essence, ESM treats an amino acid sequence as if it was a sequence of words in a sentence. Just like a sentence is made up of words that have different meanings and relationships to each other, a protein can be seen as a sequence of amino acids, each of which has different interactions with other amino acids. The masked language modeling, or in this case, the masked amino acid modeling used by ESM, involves randomly masking 15% of the amino acids in a given protein sequence, and then training the model to predict the missing amino acid based on the surrounding context. By treating protein sequences in this way, ESM models trained on over 250 million protein sequences learn an evolutionarily informed internal representation of proteins. This internal representation turns out to be highly useful for a variety of downstream applications, including protein engineering and structure prediction. It's worth emphasizing that unlike AlphaFold, which explicitly accesses evolutionary information through multiple sequence alignment, ESM implicitly learns such a representation through its large-scale pre-training. From a technical point of view, ESM was trained using PyTorch, Facebook's distributed training library Fairscale, as well as a custom Hugging Face-based tokenizer. Further, the team at Facebook AI used over 128 V100 to train models with up to 650 million parameters. Another model that has leveraged the connection between amino acid sequences and natural language is Progen. If ESM is the birth of the protein world, Progen is its GPT. Developed by Salesforce, Progen is a generative model trained with a next token, or better, next amino acid prediction objective, meaning that it is effectively trained to generate novel protein sequences. From an architectural point of view, it is a transformer decoder like GPT, unlike ESM, which is a transformer encoder like BERT. The model is trained on an enormous dataset of over 280 million protein sequences, which allowed it to learn complex patterns about the relationship between protein characteristics and amino acid sequences. Progen prefixes conditioning tags, that is, metadata about protein function, stability, and more, before protein sequences during training. This makes it possible for downstream applications to specify a set of desired characteristics at inference time. The model thus generates a protein by continuously predicting the next amino acid based both on the previous part of the sequence and on its conditioning tags. Progen 1 has up to 1.2 billion parameters, while Progen 2 has up to 6 billion parameters. Importantly, model capabilities continue to improve as model and dataset scales are increased, leading to scaling laws not too different from the ones observed in natural language processing. Both Progen models were trained using Google's JAX and Flux machine learning libraries, and the code base used to train them, that is Salesforce JAX Former, is open source and available for all to experiment with. The training itself was done on 256 TPU-v3 cores for approximately 2 weeks. Coming back to protein structure prediction rather than protein design, it's also important to discuss the now famous DeepMind Alpha2. AlphaFold2 was first introduced in 2020 and was hailed by some as a true solution to protein structure prediction. This is inaccurate, as AlphaFold2 predicts static structure when, for most downstream applications, the dynamics of a protein are essential. Further, AlphaFold2 has limited generalization, particularly when it comes to proteins such as orphan or design proteins that have very few, if any, multiple sequence alignment and thus evolutionary information available. Still, AlphaFold2 can truly be considered a giant leap forward for the field. Without going into too much detail, AlphaFold2 is characterized by two main architectural innovations. The first is the so-called EvoFormer, which is made up of two transformers, one that attends to multiple sequence alignment and one that attends to pairwise interactions between different amino acids, in a way similar to AlphaFold1's pairwise distance matrix. The two transformers communicate with each other to refine their predictions in an iterative manner. The transformer dedicated to multiple sequence alignment, in orange in the image, is tasked with identifying other evolutionarily related protein sequences that are particularly informative for the prediction at end. This informs the model's internal representation of the pairwise amino acid interactions through the connection between the two transformers. The pairwise representation transformer, light blue in the image, on the other hand, leverages triangular attention, which is a modified attention mechanism that enforces the triangle inequality, in order to allow the model to embed its pairwise representation in three-dimensional space. The two transformers of the EMFold thus iterate through the data to build an increasingly better model of the interactions within the protein, which is then used as input to AlphaFold2 structure module, which is the second important architectural innovation that characterizes it. The structure model takes the multiple sequence alignment representation and the pairwise representation produced by the EMFold as input and predicts the protein's tertiary structure. The key aspect of this part of the network is a flavor of attention called invariant point attention, which is specifically designed to work with three-dimensional structures. This attention mechanism is invariant to translation and rotations, and this fact allows the model to learn a lot more efficiently with less data. From the point of view of sample efficiency, this trick is absolutely key. If it wasn't for invariant point attention, AlphaFold2 would have to reason about every single translated or rotated structure independently. Invariant point attention thus enables the model to realize that a rotated or translated structure is still the same structure overall. From the point of view of the whole network, it's also important to note that unlike AlphaFold1, AlphaFold2 is trained in an end-to-end manner, another key feature for its success. The top right of the image has some protein structures with a comparison between ones predicted by AlphaFold2 in blue and the ones obtained through experimental, or in this case, X-ray crystallography in green. These are, of course, cherry-picked, but the image hopefully conveys what AlphaFold2 can be capable of. For some technical details, AlphaFold2 leveraged Google's JAX framework and DeepMind's own high-level machine learning library built on top of it, Haiku. The model was trained on 128 TPU-v3 cores for several weeks. Note that, importantly, this only takes into account the resources needed for the final training run. It seems fairly likely that the project as a whole leveraged orders of magnitude more compute. It's clear that this amount of computational resources goes beyond the capacity of any academic lab. In fact, very few, if any, group other than Google's DeepMind could have used so many resources. But the results clearly speak for themselves. AlphaFold2 is not highlighted in the image above, but knowing that this is a graph of the performance of each model that participated in the 2020 edition of Casp, can you guess which one of them is AlphaFold2? As mentioned earlier, AlphaFold2 represents a giant leap forward and a true breakthrough for the field as a whole. DeepMind made a great contribution to the scientific community, both by open sourcing the trained AlphaFold2 checkpoint and by releasing the AlphaFold database with over 200 million predicted protein structures. However, DeepMind did not release the JAX and Haiku-based codebase that they used to train these models. This has represented a significant limit for the wider scientific community, as any study of AlphaFold2 had to rely on the release checkpoint without the ability to have an intervention on the training process, as there was no possibility of running ablations of any kind. Thankfully, Al-Quraishi's lab at Columbia University and the extremely talented Gustav Adritz in particular have provided the research community with a solution to this problem. In particular, they have developed OpenFold, a fully trainable PyTorch-based implementation of AlphaFold. Further, they have released a set of trained model checkpoints reproducing DeepMind's results, the dataset used to train these models, as well as the results of a large amount of ablations and experiments. I'm happy to say that we have, OpenBioML and Stability, contributed in this endeavor, mainly but not only by providing some of the necessary large-scale compute. OpenFold leverages PyTorch Lightning for distributed model training, as well as DeepSpeed Stage 2 Zero Redundancy Optimizer for model and optimizer partitioning. In the experiment we've run, we utilized 8 cluster nodes of 8 A100 each for a total of 64 A100, running a large amount of ablations of 10 in parallel. We also decided to independently reproduce a full training run, which took approximately 1 month. OpenFold is a significant contribution to the open source computational biology community, since it will enable more and more groups to experiment with models like AlphaFold2. Further optimizations will also most likely enable more and more possible experiments and fast iteration ideas, as well as the exploration of many research directions in parallel. Coming back to protein language models, MetaAI recently built upon their earlier ESM models to produce ESM2, a massively scaled up protein language model with over 15 billion parameters. Just as scaling OpenAI's GPT has led to significant and at times surprising capability gains, ESM2's scaling has led to significant improvement in its internal representation of a given protein sequence. This improvement emboldened the researchers behind the model to build an AlphaFold2 inspired architecture on top of it. The resulting model is called EMFold and can be seen as an AlphaFold2-like model which leverages ESM2 embeddings rather than multiple sequence alignments to inform a structure prediction. One technical advantage of this approach is that it skips the time and storage consuming step of generating multiple sequence alignments, since ESM2 has essentially absorbed all the necessary evolutionary information through its large-scale pre-training. EMFold and approaches like it also hold promise for structure prediction of both orphan proteins, that is, proteins that are not evolutionarily related to any other, and de novo designed proteins, that is, artificially designed proteins that are completely different from any ever seen before. ESM2 was trained by MetaAI on over 800 A100 for 1 month and a half, while EMFold used 128 A100 over roughly a month. No academic research lab has access to anywhere close to this level of resources. In what is possibly the most interesting result of the paper, see the highlighted top right of the image, increasing the model size of the protein language model leads to progressive improvement in EMFold structure prediction accuracy. And this holds even for proteins that are not evolutionarily related to any other in the pre-training dataset. See the orange line in particular. ESM2 has over 15 billion parameters, but do not be surprised if someone, be it Meta or other groups, trains a far larger model relatively soon. The scaling study presented here is sufficient reason to expect renewed and significant resource investment towards developing ever larger and ever more capable protein language models. We have seen some of the most important recent developments in machine learning for protein structure prediction and design. However, machine learning also had major developments in other subfields of computational biology. Let's look at an important example in the case of genomics. As you probably already know, genomics involves studying the function, the evolution, and more generally the characteristics of genomes. But what are genomes and why are they important? A genome is the set of DNA instructions found in every cell. DNA instructions are encoded in the form of a sequence of nucleic acids, A, T, C, G in the image. The information in DNA is transferred to a messenger RNA or mRNA molecule by way of a process called transcription. Thanks to an enzyme called RNA polymerase, after that, mRNA is read by specialized organelles called ribosomes and translated in an amino acid sequence that forms a given protein. This process, which includes transcription and translation, thus gives rise to gene expression. Some parts of DNA sequences, so-called genes, lead to proteins, and those proteins perform important functions both at the level of a single cell and at the level of entire organisms. Let's take a look then at an important machine learning result in the context of gene expression prediction, starting from a sequence of DNA. Developed by DeepMind and published in 2021, ENFormer is a transformer-based model explicitly dedicated to predicting both gene expression and other important DNA-related metrics, starting from DNA sequence only. However, before we discuss ENFormer's architecture, it's important to note that one key aspect of DNA sequences is their shear length. Consider the fact that a single gene can be composed of several thousand nucleic acids, and its expression can be influenced by very distant parts of the genome, as DNA folds itself upon itself in a three-dimensional structure in a way analogous to proteins. A consequence of this fact is that machine learning models in genomics need to be able to model extremely long-range interactions between nucleic acids that are very, very far away from each other, at least in DNA sequence, but close to each other in the corresponding three-dimensional structure. This is not necessary in protein-related applications, as proteins are rarely longer than 500 amino acids. ENFormer is explicitly designed to address this particular problem. While the core of the network is a transformer, it is augmented by convolutional stem that allows the model to attend over an internal representation of over 200,000 nucleic acids. This is a key difference compared to previous work. Indeed, when coupled with the sheer power of the self-attention mechanism present in transformers, this leads to major performance improvements over the previous state-of-the-art. ENFormer is particularly effective when it comes to variant effect prediction. That is the task of predicting how gene expression varies as the DNA sequence itself is changed. This is exciting because our ability to edit genes has increased significantly over the past few years, and models like ENFormer could prove highly useful in the context of, for example, developing new gene therapies. When it comes to technical details, ENFormer was trained using TensorFlow 2.0 on over 64 TPU-v3s for several weeks. Just as in the context of AlphaFold, this only takes into account the compute power necessary to train the final model, meaning that the total compute needed for the project as a whole could very well turn out to be several order of magnitude more. As I hope I made clear with the presentation up to this point, machine learning plays an increasingly important role in computational biology and biology as a whole. And scale, be it model size or dataset size or both, plays an increasingly important, if not key role within it. Fewer and fewer groups, mostly in select industrial research labs, have access to both the engineering expertise and computational resources necessary to conduct cutting-edge research. It is for this reason that OpenBioML was founded, to provide the computational biology community with an avenue dedicated to open source and collaborative research at the intersection of machine learning and biology, as to enable researchers to take on projects that go beyond what is possible for individual research groups. Some context first. OpenBioML's main inspiration is Eleuthera.ai, the centralized research community dedicated to open source natural language processing research. Eleuthera.ai was founded in 2020 following the publication of GPT-3 in order to open source similar models. Eleuthera's results include the Pile, a foundational natural language dataset, and open source language modeling libraries like GPT-Neo and GPT-Neo-X. Further, Eleuthera.ai leveraged those libraries to train and release language models with up to 20 billion parameters, some of the most capable models available in the wild. So, what is OpenBioML? OpenBioML is essentially an Eleuthera.ai-inspired decentralized research community dedicated to machine learning in biology. It is decentralized in order to enable talented contributors from all over the world to participate in all aspects of the research process. It is collaborative to go beyond what is possible for individual researchers or individual research groups. And it conducts research in the open as to make sure that all its research output is available for the wider community to learn from and build upon. OpenBioML aims to conduct two kinds of research projects. Those include completely new research ones, such as developing entirely new techniques or innovating upon existing ones. as well as projects aiming to reproduce and open source important but unreleased models in computational biology. We consider OpenBioML a meta-scientific experiment in and of itself. Computational biology already has avenues for competitive research, and successful and famous ones as well, including the previously mentioned Casp for protein structure prediction, but it lacks an avenue for large-scale collaborative projects. OpenBioML's goal is thus that of increasing structural diversity. We are not driven by the idea that all research is better done following this approach, but by the idea that sufficiently different communities like OpenBioML will hopefully allow the wider field to take on entirely new research projects. Below is a quote from Kanjong Q's and Michael Nielsen's essay on metascience, arguing that structural diversity is a fundamental resource for science, which effectively measures and influences just how large the range of possible problems humanity as a whole can attack. OpenBioML is founded with the explicit goal of enlarging this range. The community can also act as a resource provider thanks to the support of key partners such as Stability AI that make available some of their computational resources and storage for community-sponsored projects. This means that community projects can leverage resources that only industrial research labs had access to up to now, together with tools like weights and biases for experiment tracking, Notion for project management, and much more. We hope these resources will enable the community to conduct cutting-edge research, since as we have seen, the state-of-the-art requires more and more resources. Computational biology is not alone from this point of view, as the graph below shows, and we are seeing a similar trend in most, if not all, subfields of machine learning, including computer vision, natural language processing, and many others. The community also aims to be a learning environment through both a community-organized journal club, where authors of recent research papers are invited to present their research, as well as a number of research workshops that we either have already organized or are in the process of organizing. Those include PyTorch Lightning for large-scale distributed training, weights and biases for experiment tracking, Hugging Face libraries, and many more to come. There are a few requirements for OpenBioML research projects. Those include a full commitment to open source and open science, meaning both a commitment to release all project-related research artifacts with permissive licenses, as well as a commitment to working in the open, with open project dashboard, meeting, training logs, and more. Further, OpenBioML projects should emphasize collaboration over competition, meaning that projects are expected to work on common code bases, datasets, and more. Finally, OpenBioML projects are required to be transparent over all sorts of contributions, no matter how small, and project-derived publications are expected to include a section dedicated to outlining all author-specific contributions. For a case study, one project happening in the community is DNA Diffusion, which was proposed by Luca Pinello's lab at Harvard Medical School. It involves the exploration of diffusion-based generative models for DNA sequences. Luca and his group guide the project and provide invaluable domain expertise, while many machine learning researchers and engineers in the community make significant technical contributions. The project hopefully leverages community resources to rapidly iterate on ideas and explore different directions in parallel. Thank you for listening. If you are interested in research on computational biology at scale and potentially in joining our community, consider visiting our website at openbioml.org.